talk about two parts. Firstly, about sickle cell anemia, and second part will be about antibodies. So, antibodies specifically about allergies. So, um, because we have learned for the first 2 weeks, we have learned how sickle cell anemia works and antibodies works and functions, differentiates, etc. We're gonna see how this can be developed and used in applied into in biotechnology areas. So because Justin is sick today, I'll do his part as well. Yeah, and so Modis Therapeutics, a drug development company located in Sweden, is in the process of developing a new medication which can help to alleviate the symptoms of a sickle cell anemia. And the newly developed substance called cevuparin has similar properties to the substance which is used now called heparin, but improved. So heparin and cevuparin are both a blood thinner, which means that it possesses anti-inflammatory and anti-adhesive effects. However, cevuparin, unlike heparin, does not cause spontaneous bleeding. So when the blood vessels are clotted, because of its anti-adhesive effects and anti-inflammatory effects, the clot can be removed. And however, cevuparin is also seen to have the potential to treat vaso-occlusive crisis or sickle cell crisis, which is a common complication of the sickle cell anemia, and restore the blood flow in the patient's blood vessels. However, this method can only lessen the effects that the disease inflicts on their hosts. For the alternatives, currently stem cell transplantation from bone marrow is the only way of really curing this disease, which can cause some serious side effects. But biotechnology is quickly working on the alternatives. For example, Novartis, a pharmaceutical company in Switzerland, is working with the Celexis Pharmaceutical Corporation to develop a cure to one of the complications of sickle cell anemia aforementioned, the vaso-occlusive crisis. This company aims to cure it by using a specific antibodies. So moving on to the antibodies, um, because focusing on the topic of allergies, antibodies, because as nature has also said, T cells and B cells randomly assemble different gene segments using VDJ recombinations. It is a random sequence where the gene is recombined. So um, it is really it is yet to know how it really works. So yeah, for example, like this, but this sequence VDJ recombination is also controlled and mediated by the RAG genes. However, they can be also mutated. That's why it's really hard to actually control and keep allergies under control. So what I have researched is that there's this professor from Queensland University who have considered who have researched about the memory cells, not the effector B lymphocytes, and when the allergens infiltrate our body, he found a novel way to actually control memory cells not to activate. So what he have proposed is that yeah. So this is the professor Ray Steptoe, and he said that he could wipe the memory of the T cells and in animals with gene therapy, and also desensitizing the immune system, and also this include after this he also included that the B memory cells can be also keep under control. So what he was proposing was to put artificial gene injected into a pluripotent hematopoietic stem cell, which is a stem cell for the actual all blood cells. So yeah, so he has considered that using gene editing technology such so for those of you who haven't took who aren't year 11 yet, gene editing is a technology where you could cut a segment of a gene using restriction enzymes and then replacing into a specific gene segment to control the cells. So and then they use DNA ligase to put them glue them together. So after um, because there are several cases where in the industry that pluripotent stem cells are actually cultivated, which are also called iPS. So after they are so after gene editing, they have cultivated all the stem cells, and then after that, um, the the professor's proposal is to get injected into actual blood vessel. So when the actual stem cell reaches to our body as ultimately the bone marrow, these stem cells will divide into divide themselves and also develop into the lymphocytes, so that when it eventually becomes the memory cells, they won't be activated. Uh, yes. Thank you.